So very briefly, an overview of the data that we're going to work with in these trainings. So there are two data sets here. We will work in the training, we will work with this one. It's a control sample, but I will ask you to make some exercises. Then you will work with another sample from Arabidopsis, so a bunch of sick plants. So it's Arabidopsis data, it's RNA-seq data, typical RNA-seq experiment, so unstranded. It's Illumina, it's single end. And as you will see later on, well, it's an old experiment, it's public data, so the FASTQ files are messy. Another data set that we will use mainly for the RNA-seq training is a human data set, and this is paired end data. I don't use it now because, well, you can imagine that mapping human data will take a long time because human genome is pretty big. But for the RNA-seq training, we use this one, paired end RNA-seq data from human. We will also use ChIP-seq data. ChIP-seq data from E. coli. Why? Because E. coli is so small, so these are again very small data sets. Single end, Illumina, we'll have a ChIP sample, that's the black one, and we have a control sample, and that's interesting because the control in a ChIP-seq experiment is genomic DNA cut into pieces. So the control in the ChIP-seq experiment is exactly the same as the sample that you would have in a variant analysis experiment. So with this sample, we can do everything that you would normally do on variant analysis data. But variant analysis is very often human or mouse, and that's again very hard and time-consuming to analyze. So not suited for a training. So these are the different data sets, yeah? And you can recognize them by their SRR number.